Hi everyone. So in this portion of the course, we'll be looking into tools and techniques that are used to extract information from experimental data. So these tools and techniques that we discuss here are not specific to an experimental technique and can be applied across all techniques that we study in the current course. For processing experimental image data, we'll be using an open source toolbox called ImageJ. And for processing time series signals, experimental time series signals, we'll be using functions in MATLAB. Now let's have a look at the content of this portion of the course. So we'll first look into the kind of data that we get from the experiments. So depending upon the kind of experimental technique that is used, we can either have a visual based data, which is basically an image, or we can have data from a sensor, which basically gives a time series variation of a particular parameter of our interest. For example, if we use a pressure sensor, we'll get time series variation of pressure at a particular point in the flow field. So once we have made this distinction clear, then we'll go into tools that we can use to extract out, say, parameters of our interest from the images. For example, this image on the right, it basically shows the response of a flame when it interacts with the shock wave. So during this interaction process, we might be interested in parameters such as heat release rate. So we learn about the tools that we can use to extract out this information from an image. And once we have processed this parameter over a sequence of images, we again essentially have a time series variation of this parameter. So we finally always end up with a time series variation in most of our experiments. So then after that, we look into how this time series data can be represented in using a Fourier series approximation. Here again, you look into different techniques. We look into discrete Fourier transforms. We look into fast Fourier transforms, which is basically an easier way to implement the discrete Fourier transforms. Then we look into spectrograms. After that, we'll look into the data filtering techniques to filter out the noise from the data. So experimental data is usually very noisy in nature and we need to filter them out. So here we are going to look into two commonly used techniques. One is by using FFT itself. So once we have the FFT, we can remove the noise from that using the data from FFT. Or we can use filters such as Savitsky-Golay filter or moving average filter, median filters. So we'll have an overview of these techniques. So here I have shown typical experimental images. So these are from different techniques: shadowgraphy, Schiller-and chemiluminescence. All of these are techniques that are covered in the current course. So here we can see a shadowgraphic image of a droplet that is atomizing in a high-speed flow. We can see that the droplet deforms and continuously changes its shape during the deformation, during this interaction process. And to understand this dynamics, we need to quantify the rate at which this deformation happens. And this is an information that we need to extract out from the image sequence. Similarly, if we look into the interaction of a jet flame with a blast wave, we can see that the flame lifts off and its flame area continuously changes during this interaction process. Again, we need to quantify the rate at which these changes are happening in order to study this process. Again, this is an information that we need to extract out from the image sequence. Similarly, I have another example shown here of a droplet that is impacting on a surface. So as a droplet impacts on a surface, it starts to spread radially outward. And to study the dynamics, we need to understand the rate at which this spreading is happening. And this is again an information that we need to extract out from the images. The technique that we use to extract out such information will be discussed later on in this lecture. So this is the other class of data that we have, which are basically data from sensors. So these are basically time series signals. So here we have sensors that are sensitive to specific parameters in the flow field. So if we want to measure the velocity variations, you can use a hot wire anemometer. or if you want to measure the pressure variations, you can use a piezoelectric sensor, or if you want to measure the temperature field at a specific point, you can use a thermocouple. So irrespective of what sensor we use, we always end up getting a time series signal showing the variation of this parameter at a specific location in the flow field. Usually the output of any of these sensors is a analog signal, a current or a voltage signal, and then we acquire the signal using a data acquisition unit. We'll come to that in a further slide. A very important distinction to note here is that in an image we had both, in an image sequence actually, we had both spatial and temporal data, whereas from a sensor we only get temporal variation of a parameter at a specific location in the flow field. If we want to know how this parameter varies in another location in the flow field, we need to put the sensor at that location and perform the experiment again. Whereas from an image, we can get both the spatial and temporal information simultaneously. This is a very important distinction to note. So as we saw before, an image sequence has data on both space and time, whereas data from a sensor only has time variation in the time domain. However, if we look into an image sequence, we cannot resolve all the length scales and time scales associated with an event. To understand that limit, let us look into this example over here. So here, before that, we need to understand that an image essentially consists of an array of very small units which are called as pixels. And each pixel has an intensity value and a color value associated with it. So these two images represent our satellite images of the same geographical location. However, they both have the pixels in both of them represent two different physical metric. For the image on the left, each pixel is representative of 30 meters, whereas for an image on the right, each pixel is a representative of 1 meter. So comparing the two images, we can see that there are patterns in the second image which are not resolved in the first image. That is because the length scale that is associated with these small patterns, they are lower than 30 meters, which is the pixel resolution or the spatial resolution of that image. It was visible in the second image because the length scale associated with these patterns are greater than the spatial resolution, which is 1 meter per pixel. The same concept applies even in the time domain. So here we have data that is sampled on an yearly basis, and here we have data that is sampled on a daily basis. So we can see that there are fluctuations on a daily basis, but these fluctuations won't be reflected when an yearly sample data is plotted. Again, the reason is because the time scale associated with this fluctuation is very small when compared to the spatial resolution at which the yearly data is sampled out. So in an image sequence, we have limit on both this length scale and time scale that can be resolved. That is specified in terms of the spatial resolution, which is a representative of the number of pixels for one physical unit, which is basically meter or millimeter. It is generally specified in pixels per mm or pixels per meter. And then we have temporal resolution, which is basically nothing but the time, the rate, the number of images that we have captured of the physical event in 1 second. It is usually specified in frames per second. Say if we have a frame rate of 50 fps, that means that we have captured 50 images in 1 second. That means that the time between two consecutive images is 1 divided by 50 seconds. The same concept applies even to sensor data. As we already discussed, sensor does not have spatial information, it only has data in the time domain. Although a data from a sensor is essentially analog in nature, it is usually a current signal or a voltage signal that is analog in nature. We usually use these data acquisition units to capture the data from the sensors. And these data acquisition units sample the data at specific time intervals. So there is a sampling rate. Say if we have a sampling rate of 10 kilohertz, it takes 10,000 samples from the sensor in 1 second. So the time between two consecutively sampled data is 1 divided by 10,000. So any time scale which is lower than this particular threshold cannot be resolved from that data anymore. Now that we have seen that there is a limit on the time scale and length scale that can be resolved from our experimental data, let us look at what this limit is. To understand this, we'll start off with an example. Say I have pressure fluctuations in my flow field, and these pressure fluctuations are represented by this solid lines, so solid curves. And say these pressure fluctuations have a frequency of f, and say this frequency is say 10 hertz now. Now suppose I use a pressure sensor to measure these pressure fluctuations, and the sampling rate that I set in my data acquisition unit is say fs, and say this fs by f is say 20 hertz, say 2, which basically means that fs is 20 hertz. Then the data points that we capture from the pressure sensor are these that I have shown in dots here. And if we connect these dots by using a curve, we'll be able to replicate the same periodicity as the pressure fluctuations that are happening in the flow field. Although we'll not be able to reconstruct the entire pressure fluctuations properly, since we are missing a lot of data points in between, the periodicity at least is replicated. Now suppose we reduce the sampling rate further. Say now fs by f is 1.4, which basically means that I'm sampling it at 14 hertz instead of 20 hertz. Now the data points, whatever I get, it can be traced out by a different curve, here a dotted line, and this dotted line has a completely different frequency when compared to the original pressure fluctuations, which was f. So now whatever is the frequency that I report from my pressure sensor will be completely wrong when compared to the actual event that is happening. So this actually shows what is the limit at which I can sample my data so that I can capture the physical phenomena. Say if the physical phenomena has a frequency of f, my sampling frequency should be at least 2 times of that. Now this can be said in terms of temporal resolution as well. So the temporal resolution, which is basically nothing but 1 divided by the sampling rate or 1 divided by the frames per second, should be at least should be lower than half of the lowest time scale that is associated with the problem. Again, the lowest time scale is essentially 1 divided by the frequency, the highest frequency. Here there is only one frequency, so this is simply 1 divided by f. Now the same analogy applies even in the spatial domain. To capture, say if we want to capture a length scale of L, then our spatial resolution should be lesser than L by 2. So this essentially is the Shannon-Nyquist criteria, and this is very important while setting the recording rates for our sensors and for our cameras. So we should have a general idea about what is the expected length scale and time scale, the lowest length scale and time scale of the fluid flow that we want to capture, and then accordingly set the temporal and spatial resolutions, or basically the frame rates or the pixel resolution. Now that we have distinguished the different data types, let us look into how to process each of these data types individually now. We'll start off with processing image data. Before that, let us look into what an image is. So as I had already mentioned before, image is essentially an array of a large number of subunits which are called as pixels. And the number of pixels in an image is usually specified in this format. So here if I say that I have an image which is 1020 cross 1920 cross 1080 pixels, it basically means that I have 1920 pixels along the horizontal direction and 1080 pixels along the vertical direction. Then these units, these number of pixels along each dimension essentially depends upon the camera hardware, the sensor hardware. And the images usually are of two types. They can either be a grayscale image or they can be an RGB image. In a grayscale image, each pixel has information only on the intensity, whereas in an RGB image, it has information on both the intensity and the color. We look into each of these image types individually now. So here I have a typical grayscale image. If we keep on zooming in on this grayscale image, we can see the pixels in that image. And underlying each pixel is essentially a numerical value that quantifies the intensity at that pixel. Now the intensity is specified in different ways depending upon the bit depth of the image. For example, if we have an 8-bit image, the lowest intensity is assigned a value of 0 and that has black color, whereas the highest intensity is assigned a value of 255, which is 2 to the power of 8 minus 1, and that is assigned a color of white. So if the intensity lies between this highest and the lowest, it takes up a value between 0 and 255, and each number that you see here is a representative of that level, intensity level. Similarly, if we have a 16-bit image, the lowest and the highest ranges are 0 and 2 to the power of 16 minus 1. And again, any intensity between the highest and lowest is assigned a value between these two ranges. So as I had already mentioned, here each pixel in a grayscale image only has information on the intensity. There is no information on the color. Now let us look into RGB images. So unlike in the grayscale images where the intensity level was specified only in one channel, which is a grayscale channel, in an RGB image the intensity levels are specified in three channels: red, green and blue. So if it is an 8-bit image, the intensity levels in each of these channels can be assigned the value between 0 to 255. So basically each pixel is associated with three values here, unlike in the grayscale image where it was only assigned with one value corresponding to the grayscale channel. Here it has three values corresponding to three channels. The reason why we have chosen red, green and blue as a channels to specify a color image is because these are the primary colors, and a combination of these colors in different proportions can help you replicate any other color. So because each pixel is associated with three values now, the memory that is occupied by an RGB image is much greater when compared to a grayscale image. And most of the time, in most of the image processing that we do for experimental data, most of the images are grayscale in nature. And for further processing, whatever we will be doing in the rest of this lecture, we'll be focusing on grayscale images. Okay, so now we look into how we can clean up an image data so that we can extract out features of our interest from that data. So here I'll be doing it by using an example. So here I am showing a raw image sequence of a flame that is actually propagating inside a channel. The curved object that you find moving inside this rectangular box is essentially a flame that is propagating inside a channel. It ignites at a specific location Yeah, it ignited there and then it propagated upstream, and after that it extinguished. So here I'll demonstrate our image cleaning and processing techniques using this raw image sequence. So here we might be interested in parameters such as the rate at which the flame is propagating, or the intensity profile associated with the flame during this periodic ignition extension cycle. So I'll demonstrate techniques to extract out these features, but you can always find synonymous features to extract in any problem. So whatever is the techniques that are being demonstrated, they are not limited to this particular problem, they can be applied to any problem in general. And these are the processes that are typically involved. Since our image data is usually very noisy in nature, we need to do a background subtraction to clear out the background noise. Even then, we might have to do filtering so that we can increase the signal to noise ratio again. And then we do a process of thresholding where we basically convert this grayscale image. All these are grayscale images. So this grayscale image into a binarized image where we only have 0 and 1. We don't have levels anymore from 0 to 255 all. We just have 0 and 1. So once we have a binarized image, it helps us to actually identify our object of interest. Here it's basically that curved interface that you see, the curved interface that represents the flame. So once we have our object identified, then we can trace out how that object is moving in different image sequence, and then we can extract out information such as the rate at which that object is moving, the area associated with it, or the dimensions of the object. So once we have the temporal variation of these features, we'll be able to understand the dynamics of this problem, essentially characterize the dynamics of this problem. So for this I'll be using an open source toolbox called as ImageAim, which we'll see in the next slide.